In any discussion about blood and circulatory system, you have to start with the heart. And in fact, heart is my favorite organ in the body. My research involves embryonic development of the heart, and yeah, the heart just blows my mind. So we're going to first look at sort of the gross anatomical structures of the heart before we get into how the heart works and its physiology. And so our heart has 4 chambers. 2 upper chambers called atria, and the 2 lower chambers called the ventricles. And so the right atrium, this is going to be the chamber that receives blood back from the body. And the 2 major vessels that bring blood back are the vena cava, the superior, which is bringing blood back from the head, and the inferior vena cava from the lower part of the body, including the abdomen and the thorax. The left atrium is also receiving blood back from the body, and there are 4 vessels that are bringing blood back from the lungs, and these are the pulmonary veins. Now, when you hear a pulmonary term relative to the circulatory system, understand that the oxygenation of that blood is going to be the opposite of what we usually think of for that particular vessel. So in this case, our pulmonary veins, normally in a coloring book, you would color them blue because they're veins, and we know veins are blue. But when you hear pulmonary, the colors are reversed. So our pulmonary veins are the brightest red, the richest oxygenated blood in any vessel in the body because they have just left the lungs where they gave off carbon dioxide and picked up oxygen. So they're bringing back this highly oxygenated blood to the left atrium. Now, this illustration is looking at the ventral surface of the heart. And for a lot of clinical folks, you have to begin to think of the patient. So as you look at this illustration, notice the right side of the heart is to your left, and left side is to your right, because you're looking as if you're looking through my chest, looking through the sternum at this individual's heart. We can see the blue superior vena cava in the upper left. The inferior we'll see on the reverse side, the dorsal side. And we really are only seeing a portion of the right and left ventricles. Now notice in this illustration, this is referred to as the auricle. An auricle often reminds me of your ear. And the reason that we see these terms, do you see how this is fairly flattened? This almost looks like a dog's ear or an elephant's ear. And so what I want you to more accurately refer to these structures as are the auricle of the right atrium or the auricle of the left atrium. Because the auricle is simply the roof of the atrium, and there's not a lot of muscle in the walls of the atrium, so when it's not filled with blood, it collapses down flat like you see in this illustration. That's why they refer to it as the auricle. Now also notice in this image down in the lower part, you see the bulk of what you're looking at is the right ventricle. The left ventricle you can see on the edge, but the heart is kind of not necessarily twisted, but shifted in such a way that you'll see most of the left ventricle on the dorsal side, and most of the right ventricle is going to be on the ventral surface. So again, here you can see the shift. The bulk of the left ventricle is visible here. Here you can see the opening of our inferior vena cava on the right inferior portion of the atria. Here's our left atrium, and once again we have an artist and not a biologist drawing the pictures because these should be red in color because of the oxygen content of the blood. And likewise, when blood's going to be leaving the heart, that's going to be an issue too. And in fact, here where they have the pulmonary arteries, there's that term pulmonary, we need opposite colors, and these in fact should be blue. So again, just because you see a picture on the internet, do not take it at 100% face value. Critically analyze that information you're looking at. So here we've already talked about the auricle. And so now we're going to look at the ventricles and what happens. Oxygenated blood in the left atrium is going to be pumped, we'll loosely say that, into the left ventricle, and oxygen-depleted blood in the right atrium is going to be pumped into the right ventricle. And when I say loosely, only about 20% of the blood in the atria is going to be pumped into the ventricles. 80% of the blood that's in the atria is going to get sucked down into the ventricles. And we're going to see how that works as we put all of this into motion in a couple of slides. So from the ventricles, we're going to see when the right ventricle contracts, it's going to force blood out of the right ventricle and into first a common pulmonary trunk that is going to split into a T intersection that's going to go right and left into our two pulmonary arteries, each supplying the right or the left lung. And again, these are the ones that should have been blue in the previous illustration, but they were in fact red. The left ventricle, when it contracts, blood's going to get pumped into the aorta. You can call it the trunk of the aorta, the aortic trunk. That's going to go into the aortic arch and then through the body as aorta proper, or the dorsal aorta, as many refer to the aorta. Now we're going to shift and look a little more internally at what's going on, because as our ventricles contract, much like when we looked at our axon, when we opened our sodium voltage gated channel and sodium came flooding in, where could the sodium go? Everywhere. Well, when the ventricles contract, blood is going to want to go anywhere it can, the path of least resistance. And in our heart, the path of least resistance is just to go back into the atrium. That's going to be the largest opening. So we have to ensure that blood can move from the atria to the ventricles, but it can't go back. We have to have a unidirectional valve system. And that valve system is often referred to as the Atrioventricular Valves, because that's the location where you find them, between the atria and the ventricles. And on the right side, these are comprised of three sheets of tissue, if you will, three cusps. And so that's why we call it the tricuspid valve. But understand, this tricuspid valve is not just the flaps. If it was just the flaps, blood would go backwards all day long. The flaps don't have enough structural integrity to keep the blood from going backwards. What we have connected to the flaps are these very tough cords made of collagen. They're a whole lot like tendinous material, because tendons are just collagen. So these are called chordae tendineae. So they're attached to the cusps at the valve side, and then deep in the ventricle, the chordae tendineae are attached to big bundles of cardiac muscle that are called papillary muscles. So I want you to think of our valves like the little green army men in Toy Story. The ones with the parachutes. You remember those guys? Are they still around for kids to play with? I mean, we played with them when we were kids. We loved them around this time of year, 4th of July. We'd tie firecrackers to them and throw them up and pretend they got shot down. I know we were horrible kids.
I see them in Walmart.
I think they still have some. But with the little green army men, you've got the parachute. That's going to be analogous to our cusps. The cords are the chordae tendineae, and the papillary muscle, that's the little green army man, down in the bottom of the ventricle. So when the ventricle fills with blood and it starts to contract, blood is going to be forced upward, and it's going to go underneath those cusps. They're attached all the way around the periphery. They're just free in the middle. And so as the blood gets underneath and they start to inflate and start to move backwards and start to go up toward the atria, the papillary muscles, the little green army men, they're going to contract with the same force of the blood that's moving the cusp up. They're going to pull down. And so that force is going to ensure when the cusps close, they cannot go too far backwards. And in fact, there's enough force of the blood pushing up and the papillary muscles pulling down that when the cusps close, it makes a sound. And that's what doctors are listening for with the stethoscope, the closing of the cusps. And so you have the lub-dub that they refer to it, the heartbeat. Well, the lub and the dub are made because of each set of valves. So one of our heart sounds that we're going to look at are going to be the atrioventricular valves closing. Both the right and the left close at the same time. Both are comprised of the same components. The one on the right has 3 cusps, and the one on the left is going to only have 2. So let's look at our illustration. Here we have the tricuspid valve, and why they made it in the shadows, I don't know. But on the right, this is our bicuspid. You still have the papillary muscles, you still have the chordae tendineae, and you still have the cusps. So it's designed the same way, even though you have one fewer of the cusps. Now, a lot of students have asked, well, why aren't there 3 on the left and only 2 on the right? Because the left side's going to do more work, there's going to be more pressure. The only logical rationale I can think in my head is because there's more pressure, you want fewer junctions. You want fewer places where the blood could leak. And so one line between 2 cusps is going to prevent many fewer opportunities to leak than 3 lines between 3 cusps. Does that make sense? So that's logically how I sort of wrap my head around that. Now this is a nice colorful illustration showing our atria, our ventricles. We can see our tricuspid, and the bicuspid valve is also called the mitral valve. Those atrioventricular valves prevent the backward flow of blood from the ventricles into the atria because of their design with the cusps, with the chordae tendineae, and with the papillary muscles. Now, what I want to point out here on the left side, and I kind of skipped over it on the right side, but since you're closing off the atrioventricular canals, blood can't go into the atria, blood has to get out. The right side, it's going to go into the pulmonary trunk. The left side, it's going to go into the aortic trunk. But then, after blood moves into each of these vessels, when the ventricle relaxes, it's going to create a vacuum. And the blood that was just pumped into these vessels is going to want to come backwards as well. So we have a second set of unidirectional valves in the beginning of each one of these vessels, and they're called the semilunar valves. You have the pulmonary semilunar valves, and you see here the aortic semilunar valves. Each of these is going to be comprised of 3 cusps as well, but to me they look more like pockets than cusps. There's no chordae tendineae, there's no additional muscle. And so as I compare these with pockets, imagine that my leg and my feet and my torso, this is going to be your heart. Toward my feet is the ventricle, my torso, this is going to be the aorta. So as the ventricle contracts and blood moves up my leg, I'm sliding my hand up my leg, my pocket, this is one of our cusps of our semilunar valves. As blood is moving out of the ventricle into the aorta, is my hand ever going to get dropped into that pocket? No, because it's pushing my pocket against my leg. But now, when the ventricle relaxes and the blood drops, some of that blood is going to go where? Into that cusps, inflate that cusp, and when you have all 3 of them inflate, it closes off that valve, and the blood can't move backwards. And when the blood drops and closes off the semilunar valves, there's our second heart sound. So one of the heart sounds is atrioventricular valves closing. The second or the other of the heart sounds, I forget how we've got them in our sequence, are going to be the semilunar valves. That's what makes the sounds. And so here we have a cutaway of our heart, fairly nicely illustrating a cusp on the right and on the left, our chordae tendineae and our papillary muscles. Now, I don't know that the number is quite right, but it's interesting that in this illustration, they have 3 papillary muscles for the tricuspid valve and 2 papillary muscles for the bicuspid side. I don't think that's the way it works, but it's kind of interesting that they tried to correlate it that way. Again, I think it's artistic license for this particular illustration. Now, here's an important sort of structural, functional concept that I want you to understand. The heart is comprised of cardiac muscle. And cardiac muscle cells are tied together with intercalated discs, which include gap junctions that allows them all to electrically be tied together. Until we look at the region between the atria and the ventricles. This is where we're going to attach the valves. So there's going to be a lot of tensional forces put on that tissue. So to strengthen that, there is a ring of connective tissue between the atria and the ventricles, and this is called the annulus fibrosus. And since this is connective tissue, there are no gap junctions that connect our atrial cardiac muscles to our ventricular cardiac muscles. Now, right now, you don't see a huge significance in that, but it's going to make perfect sense when we begin to look at our cardiac cycle, why that is so important and how we've overcome this disconnect and still coordinate the beating of both the atria and the ventricles at the right time. Now, here in this lower portion of our illustration, we see the cutaway. Again, we've cut away the ventral portion of the heart, exposing our atrioventricular valves, and we can see our pulmonary semilunar valves. The aortic semilunar valves are going to kind of be back behind. It's kind of underneath our mitral valve. You can't really see it in these illustrations, and it's difficult at best to see actually in the heart itself. But if you look at this top figure, this is somewhat of a cut through what might be thought of as the annulus fibrosus. You can see the tricuspid valve on the right, the bicuspid on the left. But then you can see both of our semilunar valves, and they kind of look like peace signs. And those are our 3 pocket-like cusps that are going to prevent blood from falling back into our ventricles. Now let's put the heart to work. We've got our chambers. We've got our valves in place. Now we're going to look at the cardiac cycle. This is the heartbeat. So in our cardiac cycle, we're going to have 1 beat every 0.8 of a second. Now that's on average. That gives us an individual that has a pulse about 75 beats a minute. It's not bad. So in this 0.8 of a second, 1 cardiac cycle is going to be consumed with 2 periods. And again, when you're talking about a cycle, you have to pick somewhere to start because it's just going to keep going over and over and over. So we're going to start with what we refer to as systole. Now understand the word systole means contraction. And if you only say systole, it's understood that you're talking about the ventricular contraction. And so for 0.3 of a second, that portion of our cardiac cycle is taken up with the ventricles contracting. That's both right and left. Ventricles contracting. You ever heard the term systole before? Have you heard a term that sounds like systole? What about blood pressure? Your systolic pressure. That pressure is exerted on your brachial artery in your arm because it's during the period when your heart, your ventricles are contracting and forcing the blood, and that's going to be the largest number. The same way, what do you call the smallest number? Not systolic, but diastolic pressure. That occurs in the 0.5 or half a second when your ventricles are doing what? Relaxing, and it lowers the pressure. Not down to 0. And largely what keeps the pressure from falling to 0 are our semilunar valves. It's falling backwards, so the pressure decreases, But when the valves close, your pressure can't decrease any further. So that's largely where we get our textbook blood pressure of 120 systolic over 80 diastolic. Now that's our 0.8 of a second, systole and diastole. But notice we've also got atrial contraction, atrial systole. This is going to occur in the final 1/10 of a second of ventricular relaxation. So basically our cardiac cycle are our ventricles contracting, relaxing. Contracting, relaxing. We spend more time relaxing than we do contracting. Contracting, relaxing. But now we throw in atrial contraction. So now it's like contraction, relax. Atria contracts, contraction, relax. Atria contracts, contraction, relax. See how we're just doing it right before the ventricles contract? And so in that last 1/10 of a second, the atria are going to contract and squeeze just the last little bit of blood that was in the atria into the ventricles right before they contract as a whole. So here's a cycle, just an illustration, again showing systole 0.3 of a second. Diastole is going to extend from this right hash line all the way around to this hashed line. But notice atrial systole is just this last little part. Just before the ventricles contract, the atria contract, while the ventricles are finishing their relaxation period. Now, was this in your PowerPoints that you downloaded? Okay, maybe not. This is sort of a new illustration. We're going to break down systole and diastole into subgroups. And I'll put this on D2L so that you can look at this particular slide as well. This breaks it down and gives you some of the benchmarks that are going to basically classify this particular substage, the pressures and the sounds that we get during our cardiac cycle. And so, much like we did on the board, our study habits, this is a synopsis of what's happening. If I were to ask you a cardiac cycle question on the test, this would not be sufficient to answer the question. I would need much more information, but this is how you remember everything. So I've gone ahead and provided this little bit for you. So now let's look at each of these stages of our cardiac cycle. So the first stage of systole, the first substage, is isovolumetric contraction. Now what do we know iso means? The same. So same volume, isovolumetric. But if we're contracting, how can you have the same volume? What's happening is you have the same amount of blood. You have the same amount of blood in the ventricles. But you're compressing that quantity of blood, so you're increasing the pressure. But you haven't put so much pressure on the blood that the blood moves. So this is the very beginning of contraction. But as you put the pressure on the blood and as it starts to try to move back into the atria, our AV valves close. They fill with blood, papillary muscles pull down, slap them shut, and that's our first heart sound, our S1, that's the lub. But the pressure is never going to exceed about 80 millimeters of mercury. So when we look at these 80 millimeters of mercury, you've got 80 in the aorta and the pulmonary trunk pushing against the semilunar valves. You've got increasing pressure up to about 80 millimeters of mercury in the ventricle trying to get out. So you've got the same pressure trying to get it out, trying to get in. The semilunar valves, notice, they were closed, they stay closed. So if the semilunars are closed, if the AV valves close, the blood's got nowhere to go. And so there is zero net movement of blood. That's the isovolumetric. Blood can't move. So there's our step one, isovolumetric contraction. Semilunar valves stay closed, AV valves close, and you get the first sound because the pressure doesn't exceed 80 millimeters of mercury in the ventricles. Now, as the ventricle continues to contract, that blood's going to become under more and more pressure. So we're going to exceed 80, and in fact, we're going to get up to 120 millimeters of mercury. And when we do, that's going to blow open our semilunar valves. Blood's going to rush into the aorta and the pulmonary trunk. Hence, that's our 120 systolic pressure in our brachial artery when we measure our blood pressure. Is there going to be any kind of sound when valves open? No, only when they close. So there's not going to be a sound during ejection. But I want you to notice that when the ventricles contract and blood begins to leave, only 2/3 of the blood that's in each ventricle is actually going to be ejected. This is called the stroke volume. 1/3 of the blood that's in your ventricles stay in the ventricles. Think of that as a reserve, because what you're doing right now, you're not exerting yourself very much. But if you're running for your life, your heart's going to have to work harder. It's going to have to pump more blood. So it can pump more than 2/3 because it wasn't working as hard as it could just sitting here. So that's your built-in reserve. And we refer to that 1/3 that remains as your end systolic volume, or ESV. After systole, how much blood's left in the heart? That's your end systolic volume. So in our stage 2, we've got the heart compressing, contracting, and you see our yellow arrows. They're going out the pulmonary trunk and the aorta. They're pushing against our AV valves, but they're closed. Blood cannot get back to the atria. Now when we look at diastole, that half a second, this says stage number 3 of our 5, but this is the first one of diastole. Notice it is also what? Isovolumetric. So if you remember, the first subcategory of systole and diastole is isovolumetric. Systole means contraction. Diastole means relaxation. You've already got those 2 stages memorized because the information's already there. And so as our heart begins to relax now after it's contracted, the pressure is going to drop. Our ventricle, the pressure is going to get down to 0 because we have so little blood in it. We've got a vacuum essentially. The blood's going to try to fall back into the ventricle from the pulmonary trunk and from the aorta. But when the semilunar valves close, that's going to be our S2, our second heart sound. They're going to slam shut. That's going to leave about 80 millimeters of mercury in our major vessels. That's our diastolic pressure. Now, it's also during diastole that our atria begin to fill with blood. This is blood returning from the body. It's from that 120 millimeters of mercury pushing through during systole. We're going to get this venous return, and the atria are going to start to fill with blood during our diastole step. And so here in step number 3, isovolumetric relaxation, we've got our semilunar valves closing, but notice our AV valves remain closed. So in isovolumetrics, we're going to have one set of valves closing and one set of valves remaining closed. You just have to know AVs close with contraction and semilunars close with relaxation. So again, they're just opposite of one another. Now the last part of diastole, as far as our ventricles go, this is the stage called rapid filling. Not rapid filling of the atria, that happened instead in step 3. But now our atria are filled with blood. Our ventricles have a pressure of 0, a vacuum. So now the blood in the atrium standing on our cusps of our AV valves, it's like the blood is just standing on a trap door. And that vacuum that occurs in the ventricle is going to suck the blood down past the cusp. The cusp just move out of the way. the blood falls like falling through a trap door, and that's going to rapidly fill the ventricles, but only 80% full. 20% of that volume is still in the atria. So this is why when we look at the atria, we don't say the atria pumps the blood, because really the ventricle sucks the blood out. It rapidly fills itself because of that vacuum that was created when it relaxes. You create this much larger space with no blood in it, hence the lower pressure on the inside. So that's our rapid filling, the last stage of ventricular diastole. But we still have one more thing to do before the ventricles contract again, right? At the very end of our ventricular diastole, that's when the atria contract. That's when that last 20% of the blood that is needed to fill those ventricles is squeezed in, and that is injected down into the ventricles. And when we put that last 20% in, right before the ventricles contract, that is what is considered our end diastolic volume, our EDV. End diastolic volume, how much blood is left right before we contract. End systolic volume, how much blood is left after the ventricles contract. See how you get those? And so if we take these and we put them all together, we have our 2 stages of systole. We have our 2 stages of diastole. And in this illustration, I kind of don't like how they put that 5 on there. It's just that what's happening in the atria is on top of what's going on with our ventricle. Now again, I like pictures, I like charts. This one is looking at pressure changes in the top portion. And in the lower figure, you can see systole and diastole. We look at volume changes that are happening, and there's step 1, stage 2, stage 3, stage 4, and then 5. That's our atrial systole. And then we've got our heart sounds listed at the bottom. Our S1, that's the closing of the AV valves, and S2, that's the closing of our semilunar valves. And so what we're looking over time from 0 to 0.8, that's going to be our cardiac cycle, and it repeats itself over and over and over and over and over and over every second of your life. From as soon as your heart starts to beat, and that's going to start to happen after about 3 weeks after fertilization of the egg, and it's going to beat until your last breath. And so now when we look at our summary, this probably makes a little bit more sense. This first stage lasts 0.3 of a second. What is that? What do we call this? The upper right corner. Systole. Down here for half a second. Diastole. Subcategory 1, IVC. What does that stand for? Contraction with systole. No, I mean, this is how we start to learn this stuff. Contraction, it's systole. What is the IV? We've also got an IV down here. Isovolumetric. See, we got it 1 and 3. So we also have listed less than or equal to 80 millimeters of mercury. That's our pressure in the ventricles. We've got S1 AV. What does that mean, S1 AV? S1. Heart sound 1, our AV valves closing. Notice here in isovolumetric contraction, less than 80 millimeters of mercury. S2, SL. What's SL? What's our other valve set? Semilunar valves. So you see, we got our heart sounds in isovolumetrics. We've got the same pressures related to isovolumetrics. So when you organize it and put it together like this, you're like, oh, well, that's really the same thing. It's just two different sets of valves. Then all you got to do is stick number 2 in here, ejection. Pressure inside our ventricle exceeds and gets up to 120 millimeters of mercury. Pew! Ejects the blood, but it only ejects about 2/3 or 60%. It's the stroke volume. Over here, rapid filling. We're going to suck the blood out of the atria into the ventricles. We're going to fill it with about 80% of that final volume. But we top it off with this step 5, which is sort of this orange arrow, this orange triangle there, atrial systole. So all those slides we've just walked through, summarize them here in this one picture. We abbreviate it. This is not the answer to the question of tell me the steps and the details of the cardiac cycle, but by knowing this and memorizing this picture, you're going to be able to fill in the places in the right place, in the right order. So see how that works together?